Let's start the recording, and now I gotta stop the share screen and start it. There we go. So immunity can be naturally acquired or artificially acquired. When it's naturally acquired, you get the immunity from the natural ways. You get exposed to the antigens like when you are exposed to the flu. And normally we mount an immune response that's active, meaning we respond to those antigens. But in humans, there's two cases where the humans respond passively. And that is when a fetus gets exposed to antigens because the mother comes down with the antigens, meaning she comes down with the flu. And instead of the fetus making the antibodies, because that would be active, the mother makes antibodies and then sends them over the placenta and protects the fetus. And then the mother can also make antibodies in mother's milk and then give the antibodies to the nursing child in the breast milk. Okay? Any questions about that? Immunity can also be artificially acquired when the antigens are given to you by a healthcare worker. And that's, in the active sense, is when the healthcare worker gives you a vaccine. The vaccine has the antigens to the disease, not the disease-causing organisms, and then we mount an immune response to the vaccine, and that is artificially acquired active response. Artificially acquired passive response is when the healthcare worker doesn't give you antigens, they give you already preformed antibodies to an agent that causes disease. Okay? Any question about that? So this is passive because the individual is not making the antibodies. Somebody else or some other animal made the antibodies. So let's move on to cell-mediated immunity. The cell arm of the antibody is all involving T cells. And T cells are similar to B cells, and that is once they first get activated by an antigen, they will go through clonal, what is that called? Clonal expansion and then differentiation. One T cell has one antigenic determinant on its cell membrane, and it only reacts, that T cell determinant, to one specific antigenic determinant or one specific epitope. So the T cell has many copies of that receptor, but it only binds to one epitope. And the reason why the T cells respond to so many different things is because we have a whole lot of T cells, and they each react to one specific and one different epitope. Any question about that? So when the antigens from a pathogen come into a host, and they can cause disease, but what they do is they will bind to the receptor on one of the T cells, and then that T cell will go through clonal expansion, meaning that the T cells have a T cell receptor, or a TCR, very similar to the BCRs, meaning the B cell receptor. The difference is that the T cell receptors are not antibodies. And as we discussed last time, the B cell receptor is antibody IgD, which is the first antibody that a B cell makes. And then it sticks it on its cell membrane, and that becomes the B cell receptor. Well, these T cell receptors are not antibodies. Any question about any of that? Another thing that's different for the T cells is that an antigen cannot directly bind to a T cell. Where the heck do I have a picture of that? It's not a very good picture, but what brings the antigens to the T cell is an antigen presentation cell, or APC. And for most of the times, that's a macrophage, but it can be another white blood cell. Okay, they get the antigen, they probably phagocytize the pathogen, and then they express the antigen on the cell membrane of the white blood cell or the macrophage, and then that macrophage brings the antigen to the T cell. And that's how the T cell gets stimulated. Any question about any of that? Once the T cells go through clonal expansion, they then differentiate, and some of them will become active T cells. We'll talk a little bit about that in just a little bit. They're called cytotoxic T cells. But other T cells become memory T cells, and these are long-lived cells. And the purpose of these memory T cells is that after the host throws off the pathogen, if the host is ever exposed to the pathogen later on, these memory T cells are ready, and then they mount an immune response against the pathogen. And that's similar to the memory B cells. Any question about any of that? All right. Let me remind you that the cell-mediated arm of the immune system is very good against endogenous antigens, meaning these are antigens that are associated with host cells, such as when a virus infects a host cell. The T cells are very good at finding that host cell which is infected with the virus. Or when a bacteria infects the host cell and gets inside of it, the T cells are good at finding those. They're not so good at finding freely floating antigens, meaning freely floating viruses in the blood or freely floating bacteria in the blood. The T cells aren't so good about responding against those. But they are good at finding novel antigens on cancer cells. And it's thought that about 1/3 of our human cancers are discovered by the immune system before the cancer ever develops. And then the immune system mounts an immune response against the cancer cell and kills the cancer cells. So at least 1/3 of the cancer cases in America we never see because the T cells mount an immune response against them. Any question about that? All right, so let's talk a little bit more about the different types of T cells. You can distinguish the types of T cells based upon their cell surface molecules or by what they do. And initially, we're going to divide the T cells by what they do. They're cytotoxic T cells, like this one here, and it goes out and finds other cells that shouldn't belong in the host, like a cell that's infected with a virus or a cancer cell, and then it attacks that cell, and that's the cytotoxic T cell. There are also T cells which are helper T cells, and these T cells help other cells of the immune system to mount an immune response against a pathogen. Any question about that? Well, the cytotoxic T cells, which are abbreviated T sub C for short, destroy target cells when they come into contact with them. So the cytotoxic T cell has to be touching the cell it's going to kill. And like I said, it can be a cell that's infected with a virus, or a cell that has bacteria inside of it, or a cancer cells. And how the T cell kills that cell is by one of two ways. I guess I'm talking about that now, so we will go ahead and show this picture. And then we'll come back to that little video. The cytotoxic T cell can kill the cell over here by one of two ways. It can make something, and we're just going to call these molecules, because I'm not going to use the term granzyme, which come out and they actually stimulate this cell, because it's a host cell, to enter apoptosis. And apoptosis is something that is in all of our cells, and is programmed cell death. And so the T cell is causing this host cell to undergo programmed cell death. And if you want to know, when you first performing your hand, you had cells in between the fingers. And why we have fingers is because the region in between the fingers, those cells, went under apoptosis. And that's why we have fingers, okay? The point is, all of our cells have this program for cell death, and the cytotoxic T cells can engage the cell to undergo cell death. The other way that cytotoxic T cells can kill a cell here is that it can make perforin molecules, which come on the membrane of this pathogen, and then the perforin molecules make a hole in the membrane of this cell, and then the cytoplasm can leak out of this cell. And this could be a bacteria pathogen, or a cancer cell, or a host cell that's infected. Any question about any of that? Let's watch a little video of let's see if I can get that to work. Little video of a cytotoxic. Oh shoot, let's see if another browser can do that. There was a little video of a T cell attacking a cell. Alright, this video is not going to do it either. Sorry, I've got to stop this now to... I'm not sure how to get rid of that. I've got one more browser to try. I know you're not seeing the screen. Ah, come on. Let's try Chrome. Share screen. It's not going to work. Sorry. Maybe some of you can get it to work, this video, on your own computer system, because I cannot get it to work, at least on this computer. It was a little video of a cytotoxic T cell coming up to another cell and then killing it. And you can actually see when this cell bursts its cell membrane. All right, so we talked about cytotoxic T cells. Let's talk about helper T cells. Helper T cells are cells that help other cells of the immune system respond to an antigen. In this case, we're talking about a pathogen. And as I mentioned, when the T helper cell first gets stimulated by an antigen, it has to be, the antigen has to be brought to the T helper cell by an antigen presentation cell. And most of the time that's a macrophage or their kin, another white blood cell. And then that T helper cell differentiates and will secrete cytokines, which go out and activate an inactive cytotoxic T cell, activating them. And when the cytotoxic T cells get activated, they go through clonal expansion. Some of them become a memory T cell, and then others of them become active cytotoxic T cells, and then go out and destroy any cell that has the antigen that was first brought to the T helper cell. Okay? And that's what a type 1 helper T cell does. It activates the cytotoxic T cells. Any question about that? All right. Now the type 1, already talked about them. The type helper 2 T cells, meaning a helper T cell that's of type 2, what they do is when they get activated, I don't have a picture of that, they activate the B cells and cause B cells to become activated and start producing antibodies. Now, a B cell does not need a helper T cell to activate it and start producing antibodies. An antigen can directly stimulate a B cell to become activated. And because of this, we say there are two different types of antigens. A T-independent antigen, I've got the picture, so I'm going to talk about that one first, are antigens that can directly stimulate a B cell. What these T-independent antigens have is they have many repeating subunits. And so when this antigen binds to the B cell, many BCRs, meaning B cell receptors, become stimulated. If one BCR or B cell receptor gets stimulated on a B cell, that is not enough to activate the B cell. You need many of the B cell receptors to become activated before the B cell becomes activated. Okay, going through clonal expansion and then producing antibodies. So that's why you need a specific antigen that has repeating subunits. The best example of a large antigen, and it has to be large because you have to stimulate many B cell receptors on the B cell. The best example would be a large polysaccharide that has repeating subunits, meaning repeating glucose molecules. And then that repeat can bind to many BCRs, many B cell receptors, to activate the B cell. Okay, so that's what an T-independent antigen is. A large molecule that can directly stimulate the B cell, and has to stimulate many of the B cell receptors. And so that means this molecule has to be large and have repeating subunits. And the two types of molecules that are like that are large polysaccharides. and large lipopolysaccharides. And these can be molecules found on bacterial capsules or the cell walls of bacteria. The other types of antigens are more normal molecules, like a protein. And these we call T-dependent antigens. Let's see if I can put that in the bottom. And they are a molecule that don't have repeating subunits. And so when they bind to a B cell, only one BCR will be activated, and that won't be enough to activate the B cell. So how these B cells get activated is an antigen presentation cell brings the antigen to a helper T cell that's of type 2, and then the type 2 helper T cell activates the B cells. And how they do that is they secrete cytokines, which then, instead of activating T cells, they activate the B cells. And then the B cell goes through clonal expansion and then starts producing antibodies. So that means that there are two very different types of antigens. A T-independent antigen, which is very large and has repeating subunits, and then a T-dependent antigen, which can be large, but it doesn't have repeating subunits. And these are most of the antigens that you find on viruses and on bacteria, foreign red blood cells if you're ever given a blood mismatch, and then small antigens needing a carrier molecule, such as when someone makes an immune response against penicillin G. Okay? Any question about that? We will talk about that in just a little minute. How important the T-dependent antigens are, we're going to come back to that, and we're going to look at the T cells and say that we already said you can classify them by function, but you can also classify them by their cell surface molecules. If a T cell has a cell surface molecule that we call CD4, this is primarily a helper T cell. And if the T cell has a cell surface molecule which is CD8, then this is a cytotoxic T cell. So you can divide T cells by their surface molecules. And the surface molecule is just another way of saying this cell has a different function. CD4s are helper T cells, CD8 are cytotoxic T cells. Any question about that? Now, HIV binds to helper T cells and infects them. And where does HIV bind to the helper T cell? On the CD4 molecule. And as you know, AIDS patients show a decrease in their cells that have CD4, meaning what HIV destroys in a patient is their helper T cells. What is this? That's the same picture as I'm showing here. So clearly, helper T cells are important in the immune system because an AIDS patient doesn't doesn't have a very well-developed immune system. They can mount an immune response against T-independent antigens, but they cannot mount an immune response against T-dependent antigens. And when we talk about AIDS patients, we essentially say they don't have an immune system. In reality, they can respond to T-independent antigens, but they don't have the helper T cells, so they can't respond to T-dependent antigens. And that tells you that most infections are by a T-dependent antigen. And that's why HIV patients when they become AIDS patients, their immune system is essentially shot. Any question about that? By the way, this is a helper T cell, and the red are HIV viruses binding to CD4 molecules. This T helper cell is in big trouble. Any questions? All right, I've got some animations to help you review the host defenses. The first one, I think we started that. That's the 3 lines of host defenses. And I think I will watch that because we never came back to it.
The human body has 3 basic lines of defense against...
Hold this just for a moment. And then there's a review on inflammation. And then there's a little video on B and T cells. And then there's a video on microbes and disease. It is a kid-produced video, but the science is right on, meaning their science teacher made sure that everything in the video was correct. And it's really good for a review of going over a host defenses and when you respond to an infection. But I will warn you that it has a thick British accent, so you have to get used to listening to the British accent. All right, any question about any of these reviews or links for reviews? Let's go ahead and watch this
The human
video.
body has 3 basic lines of defense against pathogens. In the first line of defense, pathogens are blocked by external barriers, such as the skin and mucous membranes. If the pathogens do enter the body, such as through a cut, white blood cells called macrophages can ingest the foreign invaders by phagocytosis. Macrophages provide the second line of defense. If the first 2 lines of defense fail to stop all of the pathogens, then the immune system, composed of both T and B lymphocytes, will specifically attack the invading cells. These cells constitute the third line of defense. T lymphocytes can react to body cells that are cancerous or have been infected with viruses. The T cell receptors recognize the antigens on the cell. This process will help the T cell in removing the affected cell from the body. B lymphocytes can recognize and react to bacterial pathogens. The B cell receptors, or antibodies, recognize the antigens on the surface of the bacteria. The human body produces B and T lymphocytes and circulates them throughout the blood and lymph. Both cell types come from bone marrow stem cells present in long bones. If a B cell is produced, it can leave the bone marrow and join the blood and lymph vessels. If the stem cell travels to the thymus, a T cell is produced, which can also join the circulatory system. B and T cells produce specific receptors using DNA rearrangements. 2 B cells may start with the same set of antigen receptor genes, but rearrange them differently. After transcription and translation, the resulting 2 B cells will have different surface receptors. Likewise, 2 T cells start with the same set of antigen receptor genes, but rearrange them differently. After transcription and translation, the resulting 2 T cells will have different surface receptors.
Okay, any questions? So that's it for host defenses 2. Let's now move on to host defenses 3. Can you guys see the screen here?
Yeah.
Thank you, Ian. All right. So this is showing you a rough outline and the major learning goals for Host Defenses 3. It's going to be looking at immunization and hypersensitivities. So be able to name the different types of vaccines and list their advantages and weaknesses, meaning there are 3 types of vaccines and they all have advantages and weaknesses. Be able to relate how B and T cells work from our earlier lesson and how they come and make vaccines, or how vaccines work and how they stimulate the B and the T cells. That would be a better way of saying it. So learn how vaccines work and how they stimulate the B and the T cells. Be able to list the 4 types of hypersensitivities and whether that type of hypersensitivity is from a B cell mediated hypersensitivity or whether it's a T cell hypersensitivity. And then be able to give an example for that type of hypersensitivity. Any questions about that? All right, then lastly, or not lastly, second to the last, be able to understand what autoimmune diseases are and be able to discuss which immunodeficiency diseases get what infections, meaning if you're immunodeficient, you'll be more likely to get a certain type of infection. All right, so this graph is showing you the value of vaccines. In about 1970, they introduced the vaccine to the mumps, and as you can see, the number of cases, I think this is in the United States, each year for the mumps before the vaccine were around 60,000 cases each year. And after the introduction of the mumps, which is not a very serious disease if it hits a child, the cases dropped precipitously, and now the cases each year, it's close, it's not quite, but it's close to 0,000 cases each year. Okay? Any question about that? Meaning the number of mumps in the United States isn't 0, but it's close to 0,000 cases each year. So it's less than, I don't know, it's less than 10,000 cases each year. Much less than 10,000 cases. It sounds like they're doing a little bit of construction here. They're supposed to start until 3, but we'll see what happens. All right, there are 3 types of vaccines, and so you have to know each one. There are attenuated live vaccines. Can you hear that banging?
A little bit.
Okay, good. You don't hear it too loud. There's attenuated live vaccines. There's inactivated killed vaccines. And there are toxoid vaccines. So those are the 3 types of vaccines. An attenuated live vaccine is where the antigens are given to a patient in a vaccine, but the vaccine is alive, meaning the antigens are on a human pathogen which can reproduce in the patient. But the virulence gene of the pathogen has been removed. And so, in theory, this pathogen is harmless. Okay? So there's our pathogen, and which one? The triangles are the virulence gene, and that comes off. And we then have a pathogen which is harmless. It has altered antigens, and it doesn't cause disease, at least not severe disease, in the person who's been immunized. Okay? And that is, in theory, how a attenuated live vaccine works. That the harmless parts of the pathogen have been removed, and then this pathogen can get in a host and reproduce, but it doesn't cause disease. And a good case in point with that would be the polio vaccine. The oral or live polio vaccine has the virulence genes removed, and then the polio virus can replicate in the patient, but it's the vaccine which replicates and the patient doesn't get disease. Now this patient has a living virus growing in them, and that was another good thing about the live attenuated vaccine, is this patient then has the live vaccine growing in them. This vaccine can move out of this patient and then infect a non-infected patient, and they will get the vaccine. Does that make sense? So when you take the oral polio vaccine, the vaccine will grow in you, and it can spread from you, and then they will get the oral polio vaccine. And that was a good thing because, as you know, in the United States, a lot of people don't bother getting the vaccines. And if we give patients a live attenuated vaccine, they will be in the population, and then they can spread that vaccine to non-vaccinated individuals. Any question about that? And that is the way the oral polio, the Salk vaccine works. And it's the way any living attenuated vaccine works. But there is a problem, and that is this organism is a living entity, although if it's a virus, I don't want to talk about whether it's living or not, but I do want to say it can reproduce. Okay? And all living things, organisms or viruses, if you want to talk about viruses, can mutate. And they can back mutate into restoring virulence. And with the live polio vaccine, which is the one best studied, there is a very low back mutation rate where the vaccine mutates back into virulence. And it's seen in about one case in 2 million people. So in 2 million people given the Salk vaccine, the vaccine can mutate to virulence and then cause polio. And polio is probably the highest case for the back mutation, meaning most live attenuated vaccines, they can mutate back to virulence, but it's going to be less than one case in 2 million people. The reason why polio is probably higher than the others is there's 6 genes that are mutated in the polio virus that make the vaccine. And if any one of them back mutate, the vaccine can be virulent. Any question about any of that? So this is a concern, and anytime you use an attenuated live vaccine in about, well, for the Salk vaccine, in about 1 case per 2 million cases, the vaccine will mutate into virulence. With the Salk vaccine, the United States doesn't use that vaccine anymore. They use the Sabin vaccine. Hopefully I've got the names right, meaning they're not using the attenuated live vaccine for polio anymore in the United States. They're only using the dead vaccine, and we'll talk about that in a moment. Certain countries, they said, okay, the first time you give a vaccine, it has to be the dead vaccine, but then the later time you give the vaccine, it can be the live attenuated vaccine. And Finland is a country that does that. And the reason is they want to remove the low rate of mutation where the vaccine can cause disease. Any question about that? All right.
Are there some pathogens that the virulence gene can't be removed?
Can you name that specific example again?
No, I was just wondering if there were some pathogens that you can't remove the virulence gene from.
I'm not aware of any where you can't remove the virulence gene. That doesn't mean that any exist. I'm just saying I'm not aware of any that you can't remove the virulence gene. Because usually what the host can respond to is more than just the virulence gene. Does that make sense? I mean, even in a virus, which they only have a handful of genes, or most like 100 genes, the host can still mount an immune response on the virus even if you remove the virulence genes. The advantage of the live attenuated vaccine is they invoke both a strong T cell response and a strong B cell response. When you give the vaccine, you only need to give one or at most a few doses of the vaccine. Like when I got the live attenuated, well, of course that's not a good example because the flu shot's usually just one shot. But when you give the oral polio vaccine, you only get it one time in the nose drops. And some vaccines, you have to give several immune shots, okay? And you don't have to give that with the live attenuated vaccine because when you're given it, the vaccine starts growing in you. And like I said, it's a strong immune response, both T and B. They are live organisms, so they can spread from the first person vaccinated to unvaccinated people. And then that can provide herd immunity, where you're vaccinating more people in the population than just the people who are getting the vaccine. Some examples of the live attenuated vaccine were the live attenuated oral polio vaccine, and I got it reversed, by the way. Sabin is the live polio vaccine. The cowpox vaccine against smallpox, that was a live attenuated vaccine. The yellow fever vaccine. Remember, herd immunity is when you protect enough people in the vaccine, and then the disease can't spread in the herd, meaning the population is safe because enough people are immunized that the disease can't spread in the population. And how many people you need to get immunized depends on the pathogen. With measles, it's very high. You need 95% of the population to be immunized, or else you don't have herd immunity, meaning the measles can spread in the population. For other diseases like COVID-19, which is spreads really well, you need 85% of the population to be vaccinated before it'll stop spreading in the population. Some things which don't spread that well, like tuberculosis, you need a much lower number, let's say 50 to 60% of the population needs to be vaccinated, and then you can stop its spread in the population. Okay? Any questions about that? All right, so that's live attenuated vaccines. Let's talk about inactivated or killed vaccines. This may have the virus or pathogen, but if it does, it is killed so that the pathogen cannot grow in you. It may not even have the pathogen. It may only have molecules or fragments of the pathogen. It requires a larger dose, and usually several boosters are required in order to get immunity. And I forgot to mention up here with the live attenuated vaccine. The immunity with a live attenuated vaccine is essentially lifelong. So if you get a live vaccine, you will be immune to that disease for essentially all of your life. There is one exception, and that is when you're elderly and if your immune system decays and becomes non-functional, well, the vaccine may not work at that point. But like I said, that's usually in the last year or two of life of people when they're sickly and their immune system goes to pot. An inactivated or killed vaccine, like I said, requires a higher dose and then several doses, and the immunity is long, like around 20 years, but it is not lifelong. Any questions about that? There have been people who have been given the inactivated killed polio vaccine, and then 20 or more years later, they came down with polio. Any questions about that? So the immunity is long, but it's not lifelong. The immune response is only humoral, meaning only the B cells respond to an inactivated killed vaccine. There is no T cell response. Some examples of a inactivated killed vaccine is the Salk inactivated polio shot, flu shots, and rabies shots. What do you notice about the naming of inactivated killed vaccines? Come on, we're not moving forward. Someone's got to guess. What do you notice about the names of an inactivated vaccine? Come on.
They're all toxin?
No, they're not toxic. Good guess. What do you notice about these three names here? The Salk shots, the flu shots, the rabies shots.
You could give them to your dog. I don't know.
They all end in the name shot. The flu shot. So we're giving this either intravenously or intramuscular. And that's why we call it a shot. Okay? So the inactivated killed vaccines, we all give them the name the shot. Like if you've ever been given the flu mist, that's the flu vaccine, and it's a live, attenuated, a live vaccine. They give it to you as a nose drop. We don't call that a shot. So the inactivated kill vaccines are usually given either intramuscularly or intravenously, and so we call it a shot. That's just my point, okay? The name tells you it's an inactivated killed vaccine. All right, the third type of vaccines are a toxoid vaccine, and here the vaccine is not meant to protect you from the pathogen. They're meant to protect you from the toxin that is produced by the pathogen. Any question about that? We haven't really talked about toxins too much, but bacteria can produce usually a protein, and then the protein can leave the bacteria, and we call them toxins. Like, oh gosh, what is that called? Botulism, the botulism toxin, okay? Corynebacterium diphtheria toxin. It's secreted from the bacteria cell. Now, there is a different toxin. We did mention endotoxin, and that's the Lipidae. It's released from the bacteria, but only when the bacteria dies. Okay? Anyways, the toxoid vaccines are meant to protect you from the toxin, from the pathogen. And what the toxoid vaccines are, they've taken the toxin and they're chemically modified, so we call it a toxoid, not a toxin, and the toxin moiety, which is this red one, gets changed. I don't know why that looks like an egg with a triangle under it and a green thing there, but the antigenic, oh I see, the antigenic determinants are now the gray part and the green part and maybe the egg on top too. That the toxin has been modified by the chemically modified, and so the toxin is no longer toxic, and we call it a toxoid. And then we inject the toxoid as a toxoid vaccine into the patient, and then the immune system reacts to the toxoid. But then when you have the pathogen that makes the toxin, the immune system will react to the toxin as well as to the toxoid. Okay, does that make sense? So the vaccine is not meant to attack the pathogen, the cell that's making the toxin, but it's made to attack the toxin. Examples of toxoid vaccines include the tetanus vaccine that is made against the toxoid of tetanus, and the diphtheria vaccine, which is made against the diphtheria toxin. Any questions about any of that? The immune response to toxoid vaccines is not strong or long-lasting. So these vaccines require several boosters during childhood, and you need a re-inoculation every 10 years or earlier. Let me think. The tetanus vaccine, yeah, you need to take it at least every 10 years. And I'm trying to remember the last time I had that, if it's been 10 years or not. And most health providers recommend a time lag of less than 10 years. I think it's something like 7 years for the tetanus vaccine. Does anyone know, anyone working in a healthcare clinic, and they know how long they're saying the tetanus vaccine should be given? Obviously, it depends on the clinician. I've heard some say 5 years. They're being very conservative. But I think most of them say something like about every 7 years. And if it's been longer than 10 years, you need the tetanus shot, because the vaccine does not last after 10 years. Your immune response has waned, meaning you're no longer protected against tetanus. All right. Like the inactivated killed vaccine, a toxoid vaccine only makes a humoral response, meaning only the B cells respond to a toxoid vaccine. There's no T cell response. Any questions about any of that? All right. I know it's getting a little loud. I think I'd like to go a little bit further, because I think at 3 o'clock I'm not going to be able to talk at all. So let's go on and talk about the 4 types of hypersensitivity. And I'll just briefly talk about this, and then tomorrow I'll try and finish this lesson. Is that right? Yeah, that should do it. So there are 4 types of hypersensitivity. And when we're talking about the immune system, the purpose of the immune system is to defend you against a pathogen. But the immune system can make you sick. And if the immune system makes you sick, we say it is an immune disease or a hypersensitivity. And so a hypersensitivity is really an allergic response, where your immune system is responding to something that it would be better if the immune system just left it alone. And so the immune system is making you sick. So the immune system can defend you, but it can also make you sick. Any questions about that? All right, so there's 4 types of hypersensitivities, and they're given really creative names. There's type 1, type 2, type 3, and type 4. Can you be more creative than that? So this is an overview looking at the types here, and you should fill this in on your own. I think actually it's just type 1, type 2, type 3, and type 4. Yep. Type 3, type 2, type 1. So I'm just asking you to fill this in, the hypersensitivity, and then whether they're B cell or T cell or not. Well, the first 3, 1, 2, 3, the hypersensitivity comes from the B cells responding. Okay? And how I know it's B cell is IgE, that's made by a B cell, IgG and IgM, also made by a B cell, IgG, and then antibodies, antigen binding to complexes. We'll talk a little bit about that when we're talking about type 3. So these are B cell, these types of hypersensitivities are responsible from the B cell. Any question about that? The 4th type of hypersensitivity is type 4, and the T cell is responsible for that hypersensitivity. And how that happens is it's the T killer cells, the TC cells, which kill target cells. Okay? The trouble is these are killing host cells, and it would be better if the cytotoxic T cells never responded to the antigen, because the immune system is just killing perfectly good host cells. Okay? Any question about any of that? All right. So let's talk about type 1 hypersensitivity, and it does have another name. You can call it the anaphylactic response. What happens in type 1 hypersensitivity, and hopefully you can see this. Let's move that a little over the way. Oh, let's move it over here. Is that in type 1 hypersensitivity, the patient initially in anaphylactic response is seeing the antigen, and we'll call that antigen an allergen because it will give rise to an allergy. And the first time the patient is exposed to this antigen, the antigen presentation cells, which are largely macrophages and their kin, will respond to this antigen and then bring the antigen to a type 2 helper T cell. And then that type 2 helper T cell will activate B cells. And this B cell will undergo clonal expansion, and most of them will become plasma cells, although there will be a few memory cells. And the plasma cell that creates the problem is a plasma cell that makes IgE. And then these IgE cells, when they're secreted by the plasma cell, the B cell, they will go into circulation and either join with a mast cell or a basophil or an eosinophil, and it can be all 3 of these cells. I think earlier I just mentioned these 2 cells. And I'm only going to talk about it happening to the mast cell. And what the IgE does is it now binds to the cell membrane of the mast cell, and then these mast cells are primed. So the first time the patient is exposed to the allergen, the antigen, the patient becomes sensitized, and on first exposure their mast cells get IgE on them, and now they're ready to respond. So the first time you have exposure to the antigen, you only prime the system. It's called sensitization. And then your mast cells are ready to respond. The trouble is, the second time you're exposed to antigens, well, let's say it's pollen in the springtime. Well, next spring you'll then be exposed to this pollen. And that pollen will come up to a mast cell which has IgE on it. And when the pollen binds to the IgE, that mast cell will then release histamines and other molecules which cause an inflammatory response. Okay? Histamine is the first molecule that starts the inflammatory response, and the other molecules just intensify and continue the inflammatory response. And then you get inflammation wherever this mast cell is, releasing the histamine. And then you get a rapid inflammatory response within minutes. If it's local, it's not usually a problem. The patient has watery eyes, red eyes, itchy eyes, maybe nasal congestion, okay? But if it's systemic and the immune response happens everywhere, meaning histamine is released into the blood and then the patient has an immune response everywhere, it can be life-threatening. The lung vessels constrict, and so the patient is hard to breathe. The blood vessels expand, and that makes it harder further to breathe. And the patient may have swelling in the throat, and then the blood vessels or the airway is constricted, and so the patient may have trouble breathing. Okay? And that can be life-threatening. Epinephrine can be given to relax the muscles and then open the airway. Any questions about type 1 hypersensitivity response? That's called anaphylactic response. All right, so the patient went to the hospital because they went into anaphylactic shock and their system totally responded, and essentially what happens is the patient, if it goes in the throat and the lungs, the patient will have problem breathing and it can be life-threatening. Okay? The trouble is, next spring, what are they going to see? They're going to see these pollen again, and they're just going to bind to the mast cells which have IgE primed on them. And then the patient will go into another anaphylactic response. This actually happened to me. I moved to Texas to go to graduate school, and the first time I was bitten by a fire ant, I simply had irritation at the site and an allergic reaction immediately at the site, but I became sensitized. And then the next time I was bitten by a fire ant, the poison from the fire ants hit my mast cells, and then I had a systemic response where my whole body got sick, and it was really bad because it lasted for about a week. And one day I'd be totally nauseous, and then the next day, what was it? don't remember. Something else would happen the next day, and then the next day I would be nauseous again. And then about a week later, my kidneys would be really inflamed. And what's happening is that the antibodies in the blood were responding to the antigen, which in this case would be the fire ant bite. And they're complexing and then precipitating out of the blood. And then my kidneys were trying to deal with it, and it put a burden on my kidney because there was so much congestion in my kidneys from the antibodies and the antigen hitting my kidneys. But then after that, I then recovered, okay? But the point is, it was systemic. The only good thing is that it wasn't ever life-threatening. It never shut down my airway and became life-threatening. And what I tried to do was rest a lot after getting bitten by a fire ant. And then when it hit my kidneys, I try and drink a lot of fluid. And that seemed to help my kidneys. All right, any question about type 1 hypersensitivity response that happens by the B cells? This one you probably should know. The first time you're primed, the time is a long time because it takes time to make the IgE. But then the next time, it's just a few minutes after you get exposed to that antigen. And those of you who have hay fever, you know this, that once you're primed, just after being exposed to hay fever, you immediately respond. Okay, just a few minutes after being exposed. And here are some common allergens. A mold, like penicillium mold. Well, this is dust mites in the house, mites that live on dust. And that's some allergen, like some pollen in the wind or something like that. And these are just some common allergies that can give rise to type 1 hypersensitivity. Any question about any of that? All right, let's move on to type 2 cytotoxic hypersensitivity response. And if it becomes too loud here, I will discontinue the lesson because it seems like they're starting to use the saw occasionally. A type 2 cytotoxic response, this hypersensitivity response, is called cytotoxic because usually cells are involved and they carry an antigen, and then antibodies are produced against the antigen, which is usually on the cells. And then the host cell destroys the cells, usually with the complement protein. I'm waiting until the saw stops. We might have to stop here. Type 2 cytotoxicity can cause many autoimmune diseases, such as a patient destroying their own red blood cells, or a patient destroying red blood cells upon a mismatch. When somebody gets a blood transfusion and they've given them the wrong red blood cells because of a mismatch. Type 2 cytotoxic hypersensitivity happens with Rh incompatibility, and that happens between the mother being Rh negative and the offspring being Rh positive. So we'll talk about it as the Rh example, but it can happen in other ways. And let me come back to type 1 hypersensitivity. I gave you an example. Allergies and then fire ants for me. What did I want to talk about? It happens from the B cells. I've lost it. I was going to say something about it. Yes, the antigen in this case may not be cells in type 1 anaphylactic response. Like for me, it was the poison from the fire ant bite. And for people who get pollen, that's pollen, which is kind of a cell. It's a special cell. But it could be dust mites, which I guess those are cells too. But it can be like a bee sting, so that would be the poison in a bee sting. This one, type 2 cytotoxic hypersensitivity response, usually happens from the antigens on a cell. So the patient is exposed to the cells and they respond to the antigens on those cells. So let's go through the RH example. The mother is RH negative and the father is RH positive. So the baby can be RH positive. If the father is RH positive and RH negative, the baby can be RH negative. But if the father is RH positive, the baby will be RH positive. And the first baby that's born that's RH positive to an RH negative mother isn't a problem. But the trouble is that blood from the RH positive baby can leave the baby and go into the mother. And that's seen as high as maybe 50% of cases, meaning pregnancy. And what can happen is there can be tearing of the placenta, and then the baby's blood, the fetal blood, can go into the mother. Or there can be other tearing of the placenta, or in, an abortion, where the doctor goes in and they remove the fetus. Well, when they do that, they can have the fetal blood leave the fetus and go into the mother. So in 20 to 50% of those cases, and this could happen during, I forgot to mention, it can happen during birth, where the fetal blood is released in birth, and then some of that blood goes into the mother. All right, any question about that? So the mother then is exposed to Rh positive blood. And then she responds by making antibodies to the Rh positive blood to the fetus. Now the first fetus, that won't be a problem because the fetus will be born before the mother's Rh or anti-Rh antibodies get made. So the first one just primes the mother. The trouble is, if she gets pregnant again, and if this child is, or fetus is Rh positive blood, the mother has antibodies which are anti-Rh antibodies. And remember, IgG can go across the placenta. And these IgG anti-Rh antibodies can then go into the fetus, and then they'll start destroying the fetal blood. And this used to be the #1 problem in pregnancy, not just in America, but worldwide. And that is that in the second pregnancy, where the fetus is Rh positive and the first fetus is Rh positive, the mother is making antibodies to destroy the fetal blood. And that can be lethal to the fetus. Okay, any question about that? Now, nowadays, it's not a problem that if the mother is Rh negative and the first fetus is Rh positive, they can give medication to prevent the mother from making anti-Rh antibodies. And so if the mother gets pregnant again and the second fetus is Rh positive, the second fetus will be fine because the mother never made the antibodies to Rh. Any question about that? All right. Let me mention that whenever the mother is Rh negative and the fetus is Rh positive, they always give the mother this medication to prevent the antibodies, regardless of what the blood type of the father is, because not all cases of the fetus are from who is the woman's husband, meaning there's almost about a 10% mismatch. Do I need to explain that any further? All right, so I think we'll stop here and I'll continue with type 3 hypersensitivity next time. Let me just say once again that type 2 and type 1, the immune system is created by the B cells because it's involving antibodies. All right? If not, we're going to stop here because it's just getting too loud. And I will not do the lab today unless there's questions. Meaning I'll start at the normal time and I'm just going to ask, are there any questions? Because it's going to be too loud.
I had a question for the unknown.
Okay. Want to ask it now?
Yeah. I narrowed my possibilities down to 2 species, but I narrowed the possibility down to 2 species, but the tests that I would perform are either one species would be positive and the other would be variable, or one species would be negative and the other one would be variable. Should I just do one of those tests, and if the other species that would be variable is the opposite of the confirmed, like if the one species, okay, so would that still be dichotomous logic?
Yes, you just do one test at a time. That's dichotomous logic. And what you're hoping for is that the variable result will be different from the other result, which depends on which test you're doing. If it's negative, you're hoping the variable will be positive. But if the variable is negative, that means that test didn't help you. Okay? And so then you go on to the next test. And you're hoping that in this case, the variable result will not be positive because the other result is positive. And so you're hoping the variable will be negative. And then you can distinguish it. So on these cases, you just have to try it and hope that you're lucky. Does that make sense?
Yeah, that makes sense. Thank you.
Okay. All right. All right. So I'm going to end it here unless there's other questions. And I'm only going to ask questions at the second part here because it'll be too loud. Okay? And we'll continue this lesson tomorrow. Okay? All right.